It is 1:40 p.m. according to my iPhone. So we'll start. Good afternoon, everyone, and welcome to this session, which is focused on immunotoxicity and immunogenicity of engineered nanomaterials. My name is Marina Dobrovolskaya, and I'm here to proudly represent the United States Nanotechnology Characterization Lab, operated by Leidos Biomedical Research for the National Cancer Institute. And I am honored to co-chair this session with Professor Janos Szebeni. We have a lot of wonderful speakers today. Each of them has a unique expertise in the given area of immunology and brings a wealth of experience. And we are looking forward to a very productive discussion today. Before we start, I would like to acknowledge Dr. Barinholtz for stimulating, initiating, and supporting organization of this symposium. And our first speaker is Dr. Janos Szebeni, who is head of Nanomedicine Research and Education Center at the Semmelweis University in Budapest, and also a world-class expert in the nanoparticle mediated complement activation.
Thank you very much, Marina. Dear colleagues, my presentation represents an extension, or actually supplements, another talk this morning by Dr. Mogimi, who was speaking about anaphylactic reactions in pigs, their cause by complement activation and macrophage activation, and he was focusing on the role of macrophages. I am going to focus on another pathway of this reaction in pigs, which is due to complement activation due to antibody formation. To put my data into a larger scheme, we are interested in the adverse effects of nanomedicines, which can be stimulatory or suppressive. And in the pathway of stimulatory adverse effects, there are acute and chronic effects. And here you can see the colored boxes show that among these stimulations, we focus on the phenomenon called pseudoallergy, and now, for the first time, on the immunogenicity of nanoparticles. The phenomenon called complement activation-related pseudoallergy was mentioned by Moin. I will touch it briefly, but the next presenter, Dr. Deji, will talk about it in more detail. I'm going to focus only on the antibody changes. And in our interest, our focus is the PEGylated liposomes, namely Doxil and Doxebo. Doxil does not need to be introduced, but Doxebo, it is a placebo Doxil, in as much as it doesn't contain the drug. It mimics completely the structure of Doxil. Well, PEG. PEG is a very important theme in our field. And I'm not sure that many of us or many colleagues recognize that there is a paradox, a huge paradox in the field of PEGylation. Namely that PEGylated liposomes have been shown to be immunogenic in rodent models, although PEG has been thought to hinder interactions with the immune system. Actually, PEG was developed initially to inhibit the interaction of nanoparticles with plasma proteins, including immune proteins. So the fact that it was found by many groups to be immunogenic is something surprising and counterintuitive. Now, there is another paradox in the field of PEGylation, namely that Doxil, which is a PEGylated liposome, is not known to be immunogenic in man. There are a huge number of patients already treated with Doxil, and yet we are not aware of any adverse immune effects of Doxil. I'll talk about it in more detail in the next slide. And yet, there is still another complication with the story of PEGylated liposomes, namely that Doxebo, the placebo Doxil, prevents complement-mediated acute infusion reactions to Doxil, which represents a passive immune suppression. The effect takes place in a matter of minutes. There's no time for active immune suppression, so we implicate a passive immune suppression. So we see PEGylation can cause immune activation, can cause immune suppression, and in people we see no immune effect. Now, a little more information, a little more background about the immunogenicity of PEGylated liposomes in murine or small animal models. These are figures from rats. Three groups, two from Holland and the group of Dr. Ishida in Japan, has been dealing with this phenomenon for a long time, and they have described in many papers that PEGylated liposomes cause the rise of IgM antibodies on a time scale of days, peaking at day 4 and then declining by about 1 week. And as a consequence of this IgM formation, the formed IgM, whenever the same liposomes are injected to rats or small animals, bind the IgM, it triggers complement activation, and that leads to the ready uptake or rapid clearance of the vesicles, which is called the ABC phenomenon. And this is a representation of the ABC phenomenon. This is the liposome injected on day 0, and then the same liposomes injected day 2, day 1, day 3, day 3, day 4, day 5, and you can see that from day 3, when the antibodies already start to rise, there is rapid elimination, and then at the top of antibody production, the clearance is almost immediate. And this is a direct evidence that antibodies play a role in this ABC phenomenon. Looking at the original PEGylation dogma, as you can see in Wikipedia, the PEGylation is qualified as attachment of PEG masking the agent or particle from immune response and thus reducing immunogenicity. So this is the official teaching about PEG. And yet, you just heard these experimental data about immunogenicity in rats. And there is almost more than a dozen PEGylated proteins already in the market, most of which can cause hypersensitivity reactions, but they are used, they are successful drugs, which implies that there is no long-term immunogenicity with these drugs. All these are PEGylated proteins, PEGylated with different chain length, different amounts of PEG on their surface. So to understand these controversies, we were initiating the experiments I am going to describe, but let me first mention about this fact that Doxil, which has been used in more than 500,000 patients by now, show no apparent adverse immune effects. And if we look at experiments in vivo, the evidence rather suggests an immune suppressive effect, namely increasing circulation time upon repeated treatments in mice, Alberto's paper, then evidence in rats of macrophage dysfunction, Storm and his group, and suppression of allergic reactions of co-administered carboplatin. This is a clinical observation, also with association of Dr. Gabizon. So there are indirect evidence that Doxil actually immune suppressive, but it is not really declared. It is not known from Doxil that it would be immune suppressive. So looking at the effects of Doxil and Doxebo on the immune system, we see this rapid phenomenon of immune antibody binding, the tachyphylaxis, which is now explained by antibody binding. The phenomenon consists of rapid reaction by injected Doxebo, and then the second time it is injected, there is no reaction, although the animal is reactive based on the reaction to zymosan. And as a consequence, we use this approach to prevent Doxil reactions, slowly infusing a small amount of Doxebo in the animals, and then treating them with bolus Doxil or Doxil infusion in the whole human equivalent dose, and there is no reaction. So in other words, with Doxebo, we can prevent the effect, the immunogenic effect of Doxil. So the experiments in which we tested these hypotheses and clarified the role of antibodies, the goal was to explore or verify immunogenicity of PEGylated liposomes, Doxil and Doxebo in pigs, to understand the role of antibodies in the immunoreactivity of Doxil, tachyphylactic effect of Doxebo, and lack of immunogenicity of Doxil in man. And the approach was we measured complement activation, reaction, and also the amount of anti-PEG, IgM and IgG in the blood of pigs before, under, and after administration of Doxebo and Doxil. About the methods, the porcine carpa model, as I mentioned, has been described in many papers, more than 60 papers over the past 15 years. It was first introduced in 1999 by this paper. There is a special edition of the European Journal of Nanomedicine, which contains a paper devoted to the porcine model. Poster 96 by Dr. Urbanic, Dr. Vasthegyi details the reaction, and the next presentation by Dr. Deji will also talk about this model. Here, just to tell that what we really look for is the rise of pulmonary arterial pressure after injection of liposomes as a measure of immune reactivity. And antibody, we measure the antibody levels by sandwich ELISA, wherein the plates are coated with either PEG, micelles, or Doxebo, and then the bound antibodies are detected by anti-porcine IgG and IgM. So this method is detailed by a poster of Dr. Kozma, poster 87. So what do we see when we pre-treat or treat the animals with Doxebo? Here is an experiment which shows the hemodynamic changes. Essentially, there was no hemodynamic change when Doxebo is introduced in the pigs in slow infusion over 20 minutes. And then, after that, the administration of the whole human dose, I mean human equivalent dose, does not cause any major reaction. What the reaction is expected here, this is heart rate. There is a minor rise of heart rate, which is unrelated to immune effects. So these animals, we refer to them like being immunized with Doxebo. And what we find as a consequence, that just like in rats, there is a huge rise of IgM starting on day 2, peaking at day 7 or 8, and then declining over 6 weeks. And if we amplify the changes, we can see that at the beginning, there is a drop of antibody level, and that at zero time, it is not, the titers are not 0. So it means that these antibodies here are natural antibodies against PEG and against the liposomes. We can't really distinguish between the two. The anti-liposome antibodies probably also recognize PEG, whereas obviously the anti-PEG antibodies recognize only PEG. We measured both, and at the moment, we can't really tell what is the difference between them. So the conclusion is that the PEGylated liposomes are immunogenic in pigs. peak at day 8 rather than day 4. The decay is longer, titers are greater, IgM and IgG responses have same kinetics, IgM response is greater, and there is initial decrease at days 1 and 2. We looked at the kinetics of antibody depletion initially during the first few minutes and found that the natural antibodies disappear in matters of minutes, and they stay at 0 or near 0 level for about 2-3 days. And from these experiments, there's not much time to explain what was done, but the conclusions is that Doxebo and Doxil bind natural anti-PEG and anti-lipid antibodies in a matter of minutes. After 5-10 minutes, the level of natural antibodies fall to near 0, regardless of the initial titer. The initial titer varies. There's substantial variation in the initial titer, but regardless, the amount of Doxebo what we inject bind these antibodies apparently. And during the time of antibody depletion, Doxil reaction is decreased or eliminated. Therefore, antibody binding explains the tachyphylactic effect or the protective effect of Doxebo against the Doxil reactions. And here, this figure shows that in immunized animals, which were immunized only with Doxebo, then administering the animals Doxebo 2 weeks later, or 4 weeks later, or 6 weeks later, the reaction becomes huge. This is not the reaction what we get from Doxebo in a naive animal, and this is what we get in an immunized animal, which implies that antibody definitely play a causal role in this reaction. And when we immunize the animals together with Doxil, there is no reaction, which implies that Doxil was immune suppressive. It prevented the formation, it prevented the immunogenicity of Doxebo. So essentially, this finding, this experiment explains why Doxil is not immunogenic in men, although the coat, the liposome membrane might be immunogenic, at least in some people. The presence of the doxorubicin, the cytotoxic agent, prevents this immune activation. And this figure shows the anaphylactic reaction, what we observe in pigs treated with Doxebo during the time of immunization. Huge reaction leading to death in a matter of minutes. And this particular animal was saved by epinephrine. But you can see that within a minute, the blood pressure drops to 0 and remains there unless the animal is salvaged. This is a very huge, very marked anaphylaxis. So concluding, pig blood contains low and variable amounts of natural anti-PEG and anti-liposome IgM and IgG antibodies, which explains, at least in part, the carpogenic effect of Doxil. And here, let me emphasize again that what Dr. Mogimi told in the morning was the role of PIM cells, the macrophages, and actually macrophages are in the pathway of this reaction. So the two statements are not contradicting each other, but actually complement each other. The natural antibodies can be depleted by a small dose of Doxebo or doxorubicin-free placebo Doxil, which may explain the protective effect of Doxebo. Doxebo itself is highly immunogenic, leading to massive production of anti-PEG and anti-IgM, IgG. During antibody induction, the CARPA caused by liposomes lead to anaphylactic shock. The immunogenicity of Doxebo and consequent anaphylactic reactivity can be prevented by the human equivalent dose of Doxil, which explains the absence of immunogenicity of Doxil in patients. Perhaps most importantly, these experiments highlight the unique utility of pigs as a functional model of nanoparticle immunogenicity leading to anaphylaxis. We use the pig as a model for CARPA until now, but this is a new challenge for us to use the model for studying immunogenicity. And I came to the end, acknowledging my colleagues and all others who were taking part in these experiments. Thank you. So, if someone has a question,
Janos, what do we know about anti-PEG antibodies in humans, in cancer patients?
it was described by many authors that about at least 30% of people today, nowadays, contain anti-PEG antibodies in their blood. Interestingly, there was another paper like 10 years ago, and then the ratio, the portion was a few percent. And nobody knows whether this increase is real or an artifact due to different techniques. But yes, humans do have anti-PEG antibodies. Cancer patients, obviously, I mean, they are not different in that respect. And that might be at least partly explained why some people react to Doxil or PEGylated liposomes, but not everybody.
Would you recommend testing for these antibodies before starting treatment with PEGylated liposomes?
Obviously, yes. Based on these results, measuring anti-PEG IgM could be predictive of the reaction.
There is a poster by him, we just saw it, about in a very large number, I think number 80 if I remember well, measuring level of IgG and IgM in about 1,500 people. They are naive people, not treated with anyone, and I recommend you to see the poster. It's really amazing that, as Janos said, even higher level, 40%, and women have more higher level than men. And talk with him about the explanation. It's very interesting.
It also raises the question. It raises the question: why do we have anti-PEG antibodies in our blood? And one explanation is that we are exposed to a lot of PEGylated stuff in hygiene and in our food. But what is really surprising is that we also have the natural antibodies against PEG in pigs, which are not supposed to have all these fancy PEGylated food.
We don't know. You have to look at the food of the pig, of the pig. You have to look at the pig, at the food. I mean, almost, it's amazing the number of food products that contain PEG, if you check. It's really amazing. So I think it's a very cheap ingredient. It helps to make whatever, when you feed them with something artificial, the PEG will be very helpful. We have to check it. We don't know.
So this is a wonderful discussion just for the sake of time, so we stay within the agenda. We will have, at the end of this session, we will have more expanded discussion, but there is a question on the back. So I would like to share questions so that we allow more people asking.
Janos, the IgG in a naive animal is a little bit suspicious. I just wondered, have they been immunized for anything else? Are they truly naive pigs?
Well, they are truly naive pigs. The IgG level, the natural IgG is much lower. The titers are much lower, but definitely present. And there is a technical issue in that the anti-IgG, the anti-porcine IgG may cross-react with the with the porcine IgM. So the level of IgG could be actually underestimated because of the cross-reaction of the detecting antibody with the IgM. And that was not solved, but in any way, we could underestimate the IgG rather than overestimate. And I do not know why those data appear suspicious for you.
In human, if I remember well the data, they're about equivalent.
Anyone else? Any other questions?
You may wonder about the Ig the IgG against the PEG. But what I always find very interesting is why are there IgGs against phospholipids? I mean, we are full of phospholipids. So why is the IgG there? I mean, this is a story I know that Jesse wants to say something about it, but this is around for 30 years, I think. And everybody's coming up with these antibodies against phospholipids, but we consist of phospholipids, so what are they doing there?
Well, I think it's a really interesting thing, interesting point. You know, I remember when I did, as a hobby, I was teaching kind of the introduction to gerontology. And one of the things we see in human beings, that with age, the level of our antiphospholipid antibodies is going up. And I think the Carl Alving that worked with, that Janos worked with for many years, and there is another one before him, notified that there is various levels of anti-IgM and anti-IgG. And I remember when we developed a vaccine for influenza based on phospholipid, on DMPC and DMPG, we found that this vaccine induced increase in the level of antibodies, antiphospholipid antibodies. You know, it's a routine test now because it's problem number one in women, and it really a major issue of loss of baby. About 1% of, I think the birth ended not very well because of antiphospholipid antibodies. I don't remember the name of the disease anymore. It was long ago. Yeah, no, not lupus. Lupus is another story. Lupus is cardiolipin. It's another story. No. What? Yeah. Okay. So they are, but in most of the cases, it doesn't do us any bad. I mean, that's the point, the presence of the antibodies. So it's a kind of naive. But antibody against cholesterol, if I remember well, may be well involved in the complement activation.
Actually, there are more people anti-cholesterol antibodies than anti-phospholipid.
That's right.
And
also increase very much with aging. And also, people were looking of it in relevance to atherosclerosis, and it's still a very controversial issue.
yes, and there were attempts to make an anti-cholesterol vaccine on the basis of anti-cholesterol antibodies.